Hello everyone, welcome back to the Pharmacy India. So in this session we are going to discuss most important MCQs questions of carbohydrate and lipids, which will be very important for your MRB Pharmacist exam. So without any delay, let's get started with our first question. But before moving on to the question, make sure to download Pharmacy India mobile app from Google Play Store so that you can join any type of test series for your practice, and also you can join any courses from Pharmacy India mobile app. So let's get started with our first question. Question is, which of the following enzyme catalyzes an irreversible step in glycolysis and is a key regulatory point? So from the given options you have to choose that enzyme which catalyzes irreversible step. Hexokinase, phosphoglucose isomerase, aldolase, or glyceraldehyde 3-phosphate dehydrogenase. The correct option is hexokinase. What is the function of hexokinase in glycolysis? Is it converts glucose into glucose 6-phosphate. Glucose 6-phosphate, which is the first step in glycolysis. Clear? It is the first step in glycolysis, and it is a very important enzyme in the process of glycolysis, which is also known as EMP pathway, Embden-Meyerhof pathway. Clear? Hexokinase phosphorylates glucose to form G6P, trapping glucose inside the cell. This irreversible step controls the entry of glucose into glycolysis. What is glycolysis? Glycolysis is breakdown of glucose. Breakdown of glucose. Clear? So the correct option is A, hexokinase. The Cori cycle involves the transport of which molecule from muscle to the liver for gluconeogenesis? Before coming to the question, you should know about the Cori cycle. Cori cycle is a type of metabolic cycle in which lactic acids convert into glucose and pyruvic acid converts into lactic acid. Clear? This is a Cori cycle which continuously functions in human body. Alanine, glycerol, lactate or pyruvate. Correct option is lactic acid, lactate, which forms in liver for gluconeogenesis. Now what is gluconeogenesis? Formation of new glucose. And why lactic acid form? Because it ultimately converts into glucose into liver. Clear? It cannot be stored in this form, lactic acid form. Next question. Deficiency in G6P primarily affect which metabolic process in the liver, leading to hypoglycemia in glycogen storage disease type 1, which is also known as von Gerg disease, which mainly affects your liver. Glycogenolysis, gluconeogenesis and release of free glucose or glycolysis. Basically, glucose 6-phosphatase is a very important enzyme in the process of gluconeogenesis. Clear? Gluconeogenesis. With the help of glucose 6-phosphatase, we convert glucose 6-phosphate into glucose 6-phosphate into glucose. Clear? So the correct option is C, gluconeogenesis. And by the deficiency of glucose 6-phosphatase, there is a disease, glycogen storage disease, which is called von Gerg disease, that affects your liver. Clear? So the correct option is gluconeogenesis. G6P convert glucose 6-phosphate into free glucose. A deficiency prevents the final step of both glycogenolysis and gluconeogenesis, which cause hypoglycemia. And what is hypoglycemia? Hypoglycemia is a term which refers to low blood sugar level. Blood sugar level. Clear? Next. Which of the following is a major function of hexose monophosphate pathway or pentose phosphate pathway? It is the same thing. Production of ATP. No, this is not the function of HMP shunt. Generation of NADPH for fatty acid synthesis and ribose 5-phosphate for nucleotide biosynthesis. Nucleotide means for the formation of ribonucleic acid and deoxyribonucleic acid. Oxidation of pyruvate to acetyl coenzyme A, wrong. Breakdown of glycogen to glucose. No, this is the process of glycogenolysis. Correct option is second. Generation of NADPH means nicotinamide adenine dinucleotide phosphate hydrogen for the synthesis of fatty acid, and also ribose 5-phosphate for nucleotide biosynthesis. That means for the formation of RNA and DNA. This is the main function of HMP shunt. Clear? So the correct option is B. The branching enzyme in glycogenesis is responsible for the formation of which type of glycosidic bond in glycogen? Which bond is present in glycogen? This is the simplest question among all the questions. Alpha 1 for glycosidic bond, alpha 1 6 glycosidic bond, beta 1 4 glycosidic bond, or beta 1 6 glycosidic bond. Basically, glycogen is a polysaccharide, homopolysaccharide, which consists of glucose units. Clear? Glucose units. And they attached with each other like this. Clear? Like this. So the correct option is alpha 1 6 glycosidic bond. Alpha 1 6 glycosidic bond. Glucose present in the glycogen in the form of amylopectin. Amylopectin. Amylopectin is a branched structure made up of glucose units. So the correct option is alpha 1 6 glycosidic bond. What is the meaning of 1 6? See, this is glucose number 1, and this is glucose number 2. Clear? And this is glucose number 3. So basically there is a bonding between this and this. Clear? This is carbon number 1 and this is carbon number 6. So the bonding between carbon number 1 and carbon number 6 forms alpha 1 6 glycosidic bond. Which enzyme catalyzes the rate limiting step in fatty acid synthesis? What is the meaning of rate limiting step or rate limiting enzyme? Those reactions or those enzymes which controls the speed, the rate, the velocity of reaction are known as rate limiting steps or rate limiting enzymes. Clear? So in the process of fatty acid synthesis, acetyl coenzyme A carboxylase is the rate limiting enzyme. Clear? Which controls the whole speed of, which controls the speed or rate or velocity of the whole reaction. Carnitine is essential for the transport of long chain fatty acid into which cellular compartment for beta oxidation? Carnitine is a type of transport protein, or you can say carrier protein, which is responsible for carrying long chain fatty acids into mitochondrial matrix for the beta oxidation of fatty acid. This is the second step in beta oxidation of fatty acid, where carnitine transport fatty acid, long chain fatty acid into mitochondrial matrix. Correct option is C. Long chain fatty acids are first activated into the cytosol into acyl coenzyme A, and then this acyl coenzyme A transported into mitochondrial matrix with the help of carnitine. Clear? This is the second step in beta oxidation of fatty acid. The process of beta oxidation of a saturated fatty acid with an even number of carbon atoms yield which primary end product? Glucose, pyruvate, acetyl coenzyme A, NADH and FADH2, or lactate. Basically, pyruvate, glucose and lactate, these are the end products in the formation or breakdown of glucose or glycogen or lactic acid. So the correct option is C. Acetyl coenzyme A, NADH or FADH2. These are the end products which forms at the end of beta oxidation of saturated fatty acid with an even number of carbon atoms. Clear? NADH is nicotinamide nicotinamide, adenine, dinucleotide, hydrogen, and FADH2 stands for flavin, adenine, dinucleotide, hydrogen. Clear? This is FADH and this is NADH. Ketone bodies are synthesized in the liver mitochondria from excess acetyl coenzyme A, derived primarily from the oxidation of glucose, amino acid, fatty acids or lactate. Basically, ketone bodies are considered as fuel source if there is a deficiency of carbohydrate, lipid and other nutrients in the body. Now, from which of the following component ketone bodies are synthesized in the liver mitochondria? Glucose, amino acid, fatty acid or lactate? Correct option is fatty acid, because first fatty acids converts into acetyl coenzyme A by the process of beta oxidation, acetyl coenzyme A, and with the help of acetyl coenzyme A there is a process which is known as ketogenesis, through which we form ketone bodies. Three types of ketone bodies in the human body which act as the fuel source. During periods of low carbohydrate availability, fatty acid oxidation produces excess acetyl coenzyme A in the liver, which is then used to synthesize ketone bodies as an alternate fuel source, as I have told you earlier. Which of the following ketone bodies can undergo spontaneous non-enzymatic decarboxylation? Non-enzymatic decarboxylation means decarboxylation without any type of enzyme. Clear? Beta hydroxybutyrate, acetoacetate to acetone, acetyl coenzyme A, succinyl coenzyme A. Succinyl coenzyme A is not a type of ketone body, so it will be eliminated. Correct option should be acetoacetate to acetone. Clear? This is a type of reaction in which acetoacetate decarboxylation into acetone without any type of enzyme. Clear? So the correct option is B, acetoacetate to acetone. Basically, our body contains three types of ketone body. First is acetoacetate, acetoacetate. Second is acetone, and third is beta hydroxybutyrate, Acetyl coenzyme is not a ketone body. It is a type of precursor which is used to form ketone bodies. Clear? Next. In the fed state, which hormone promote glycogen synthesis in the liver and muscles? Glucagon, epinephrine, glucocorticoid or insulin? Basically, for the formation of glycogen in the liver and muscle, we use insulin, because insulin is a hypoglycemic hormone, and it is used to lower down blood glucose level and helps to convert glucose into glycogen into liver and muscles. After a meal, elevated blood glucose level trigger insulin release from the beta cells of pancreas, and with the help of this insulin, glycogen synthase and enhances glucose uptake, which favors glycogen storage into the liver and muscles. During fasting, which hormone stimulates glycogenolysis in the liver, leading to the release of glucose into the bloodstream? Now, insulin is a hypoglycemic hormone, but there is also another hormone which acts exactly opposite to insulin, which is called glucagon. It is a type of hyperglycemic hormone. Why? Because it helps in breakdown of glycogen back to glucose. Clear? Back to the glucose. So the correct option is A, glucagon, which is released from the alpha cells of pancreas. Clear? Alpha cells of pancreas. Atherosclerosis, fatty acid and hypercholesterolemia are associated with defective metabolism of carbohydrate. No. Lipids, yes. If lipids cannot be metabolized into the body, it results in atherosclerosis, means LDL accumulation in between the arteries or veins. Clear? This is low density lipoprotein which leads to atherosclerosis. Fatty liver means inflammation of liver due to the accumulation of fatty tissues, and hypercholesterolemia means if the level of cholesterol reaches above 200, clear, that considered as hypercholesterolemia, and these are the diseases which are associated with the defective metabolism of lipid. Which pathway provide UDP glucuronic acid? What is UDP? UDP is uridine diphosphate. ठीक है, uridine diphosphate, which is essential for the conjugation of bilirubin. And what is bilirubin? Bile pigment. Bile pigment. And detoxification of certain drugs. Glycolysis, citric acid cycle, uronic acid pathway, or HMP shunt. Obviously, correct option is C, uronic acid pathway, which is used to produce UDP glucuronic acid, and UDP glucuronic acid is very important for the conjugation of bilirubin and detoxification of certain drugs. The uronic acid pathway converts glucose into UDP glucuronic acid, a molecule crucial for conjugating bilirubin and other toxins, thereby enhancing their solubility for excretion from the body. Glycogenolysis involves the cleavage of alpha-1,4 glycosidic bond by which enzyme producing glucose 1-phosphate. The question is asking about the enzyme which is help to convert alpha-1,4 glycosidic bond and help to produce glucose 1-phosphate. 1-phosphate. Your options are glycogen synthase, debranching enzyme, glycogen phosphorylase and phosphoglucomutase. We have to cleave alpha-1,4 glycosidic bond, so we have to use glycogen phosphorylase, which is the key enzyme, key regulatory enzyme in the process of glycogenolysis. What is glycogenolysis? Breakdown of glycogen. Clear? To form glucose. Glycogen phosphorylase catalyzes the breakdown of glycogen by cleaving alpha-1,4 glycosidic bond, and it is used to yield glucose 1-phosphate, which can then be converted to J6P and then glucose. Next question. The conversion of pyruvate to acetyl coenzyme A is catalyzed by the pyruvate dehydrogenase complex and is an important link between glycogenolysis, gluconeogenesis and glycolysis and the citric acid cycle or fatty acid synthesis and beta oxidation. Conversion of pyruvate to acetyl coenzyme A is a connecting link between glycolysis and citric acid cycle. Why? Because in citric acid cycle, acetyl coenzyme A acts as the precursor, which is used to convert into citrate. Clear? And acetyl coenzyme A is used in citric acid cycle. Clear? So the correct option should be C. The pyruvate dehydrogenase complex converts pyruvate from glycolysis into acetyl coenzyme A, which then enters the citric acid cycle, and it serves as the crucial metabolic bridge between anaerobic and aerobic energy production. Clear? Which of the following is a non-carbohydrate precursor that can be used in gluconeogenesis? Non-carbohydrate precursor, okay. Acetyl coenzyme A, fatty acid with an even number of carbon atom, glycerol or glycogen. glycerol and glycogen. These should be eliminated from the options because question is asking about non-carbohydrate precursor. Option is glycerol because glycerol is a lipid. Clear? Glycerol is a lipid and it is used in the process of gluconeogenesis, means formation of new glucose. Clear? Glycerol derived from the breakdown of triglyceride can be converted into dihydroxyacetone phosphate, that means DHAP, and then utilized in gluconeogenesis to form glucose. The enzyme phosphofructokinase in glycolysis is allosterically inhibited by phosphofructokinase PFK1 is the rate limiting enzyme in glycolysis. If we controls the activity of PFK, we can ultimately or eventually controls the rate of glycolysis. AMP means adenosine monophosphate, fructose 2,6-bisphosphate, ATP and citrate or insulin. ATP and citrate are the allosterical inhibitors of PFK1. Clear? So the correct option is adenosine triphosphate and citric acid. Second last question. Which of the following lipoprotein is primarily responsible for the transport of cholesterol from peripheral tissues back to the liver? Peripheral tissues to the liver for metabolism. Clear? This is very important for the metabolism. Clear? Correct option is high density lipoprotein, which is also known as good cholesterol. Good cholesterol. And why it is known as good cholesterol? Because of this activity. Clear? HDL facilitate reverse cholesterol transport by collecting cholesterol from the peripheral tissues and transport it to the liver for further metabolism and help to prevent accumulation of excess cholesterol or lipids in the body. This function is important for maintaining cholesterol balance and reducing atherosclerosis risk. Clear? And last question. Deficiencies in enzyme of glycogen metabolism can result in a group of inherited disorders known as glycogen storage disease. For example, McArdle disease, von Gogh disease, or there are so many diseases which are associated with the deficiency of enzyme which are involved in glycogen metabolism. Clear? So the correct option is glycogen storage disease. Clear? So these are the very important questions of carbohydrate and lipids. I hope these questions are very much clear to you. I'll see you in next class. Till then, stay tuned with Pharmacy India. Thank you.